Hope you all had a great holidays and restful break. So thanks again for joining me today in this webinar, BioRender 101, Improving Communications with Visuals. My name is Jerry. My goal today is to provide you with a foretaste of what our platform can empower you to do in improving your science communication. As for our agenda today, I'll be walking through a bit of BioRender's background and put most of the attention on a concise show and tell live demo. If you have any questions at all at any point in the webinar, please feel free to ask in our Zoom webinar chat, or you can try the question and answer, feature of Zoom webinars, and I'll try my best to answer them as you ask. But before we get started, I'd like to just do a very quick Zoom poll with everyone to get a better sense of your background, what you're doing, and whether or not you've used BioRender before. So let me just launch this poll here. You'll probably see a poll window pop up where you can answer it. So please do so. Thank you. I'll just leave it up for another 10 or 15 seconds or so, just to get all the votes. Okay, cool. So it looks like we have a mix primarily of scientists who have not used BioRender before with a couple who have. And in terms of backgrounds, we have primarily with us today a academic audience of graduate studies students as well as PIs. Thanks, everyone. So just to, I guess, reassure everyone for today's for today's webinar, hopefully the way I presented it will be helpful both for completely new interested users for BioRender platform, as well as for anyone who has experience with our platform, as I'll be walking through functionalities, but also design tips for creating sensible, clear figures. Okay. So I guess just to get started, a little bit about myself. My name is Jerry. I come from a life science background studying biochemistry in undergrad and graduate school. I've also had a passion for visualization and art since I was a kid. So I went on to pursue this interdisciplinary degree in biomedical communications at the University of Toronto in Canada. This gave me the skill set needed to illustrate, animate, and redesign professional quality scientific and medical content for better communication. However, I did over time quickly realize that something more was needed to bring this service, or I guess this need to enhance science communication to the scientific community at large. And because of that, I ended up joining BioRender. So how did BioRender come to be? Our BioRender CEO, Shiz Aoki, and its co-founder came from a similar program as myself in the US. Her professional career led her to become a lead science illustrator at National Geographic for a decade. On the side, she also worked extensively with scientists and labs and popular academic journals such as Science and Nature. So she identified that most scientists and labs don't have the budget or the resources or the connections to collaborate with professional science illustrators to visualize their science. Now, unfortunately, what ended up happening then, as many of you might already be aware, is that in the life science community, most scientists end up using PowerPoint or something similar, spending hours to try to make an illustration to articulate their findings. And this led everyone to come up with dozens of different ways to show essentially the same pathways or the same reactions or methodologies with different degrees of success. for instance, right here, it's just a tiny fraction of attempts we see of scientists visualizing a foundational immunology concept of cells called antigen presenting cells, presenting antigens to T cells. So the co-founders of BioRender then asked a key question: what would happen if we, as science illustrators, somehow made all the graphical elements involved in scientific figures standardized and freely available to scientists? Would the scientific community be able to put them together to make their own compelling, clear illustrations and figures? And that was the seed of how BioRender got started. So what is BioRender now? It's a web-based program and platform designed specifically for scientists and researchers. We're a visual asset library with many thousands of customizable life science icons and templates across more than 30 fields, plus extremely versatile dynamic arrows and shape drawing tools. The functionality is all drag and drop. You can make illustrations up to 50 times faster than in PowerPoint. And the best part is you don't need a design background to make clear, elegant figures. Now, BioRender has been used most frequently by our user base for the 3 Ps, we will call them, publications, presentations, as well as posters. But our users are finding tons of other uses as listed here, as well as more new interesting ones, like for patents, YouTube educational videos, course materials, etc., etc. Here's just a short clip of how the software works at a glance. Very, very abbreviated, but of course, I will go over this in a lot more depth during the demo to come. I also mentioned that we have templates in our library. So what I mean by templates essentially is, at the moment, figure templates for various different figures across different fields for different purposes. These are just a couple of examples here, covering pathways, clinical figures, experimental methods and protocols, graphs, timelines, etc. We're adding more every day. And in fact, I think currently we have a few hundred templates, but in 2020, I'm actually involved in a new initiative at BioRender to really ramp that up and really expand our template library further. They're all fully customizable from ground up. Essentially, each of them function as a BioRender illustration, but with proper kind of design sensibilities built in. And some of them have placeholder text, others have established text that you can just modify. And the main goal is that they're designed so it's easy to start if you don't like the idea of starting from a blank canvas. And yeah, so at BioRender, our goal is to give every scientist both the tools as well as the practical knowledge to visually communicate their research. With that in mind, my aim today, as I initially mentioned in our webinar today, is to demonstrate with everyone the capability of our platform, as well as provide some general practical knowledge of designing good figures. On that note, let's jump into BioRender and have a go then. I'm going to switch over. There we go. Okay, so here we are. We are in the gallery page of a BioRender account, just a demo account I set up for this webinar. As you can see here, just a bit of orientation. Under here is an area of file management, and above you can choose to start from a blank illustration or from some of our templates. You can see a lot more of the templates by clicking see all templates here or going to BioRender templates at the top. Oh, actually, before I continue, Sung Ming asked a great question. Asked, should I add reference like from BioRender once I use any figures from BioRender? That's a great question. So essentially what we request and ask everyone to do, if you are making a figure from BioRender and let's say you're including it in a thesis or publication or on a poster, that we'd like if you can credit us either in acknowledgement section if there is one, or at the end of the figure captions, essentially created in BioRender.com, something like that. Hope that answers your question. Okay, so jumping back to gallery here. Let's say today I'll start from blank illustration. You can see a little sneak peek of roughly what we're going to be building. So I click that. I'm going to call it webinar figure 2020-1018. And in terms of illustration size, I'll probably just stick with slide size. You can choose a size that fits your needs here, but essentially you can adjust the illustration size at any point when you're editing the figure later on. So create illustration. And here we are. We arrived at the canvas view of BioRender. And just to orientate everyone a little bit, on the left here, you can navigate through the icon library, either through menu systems or through the search bar. You can access templates here as well, if you'd like. Instead of outside in the gallery, you can go into Canvas view and basically apply or add BioRender figure templates onto the canvas itself. There's a few other things I'll walk through later on. And across the middle here, middle top here is the toolbar, which is contextual, as in whatever you select on the canvas, the toolbar will adjust to show you the features and the functionalities that you can manipulate it with. On the right hand side here, we have zoom, as well as a few other features such as showing grid and ruler. Actually, I'll keep ruler on. It's actually one of our newer features that we're pretty excited to show off. So I'll keep it on for now. And at the bottom here, if over time you'd like to become more accustomed to using BioRender faster, you can access keyboard shortcuts here and start trying some of these out and see if they speed up your process of building figures. And of course, you can reach live chat with our excellent customer/user support team, or access our FAQ. So with that, having gone through that, let's start our figure creation demo today by first thinking about what we want to make. So I'm thinking of potentially a figure where I want to show, going back to what I mentioned in the intro, maybe a antigen presenting cell presenting a foreign antigen to a T cell. And I want to design the figure in a way so that the cell comes in from the top left and somehow presents the antigen to the T cell at the bottom right. And then we can kind of go through maybe a pathway, a very abstracted pathway through the T cell. And that's hopefully going to show a bunch of different functionalities as well as design decisions. Okay, just to get started then, I'm going to look for some cell icons. So I think I want the cells to be fairly big, so I'll actually search for phospholipid bilayers. Something like that, a membrane, plasma membrane. And I like this one, so what I'll do is just click and hold my left click down and drag onto the canvas and release. There we go. And then I'm actually going to zoom out a little bit more so that I can resize this using the bounding box and rotate it here. And just getting it to fit nicely. Something like that. And then I think for the T cell, I want it to be blue. So I'm going to click icon color here in the toolbar, and let's try this blue here. It's good. There we go. Okay, with that put in, I'm gonna maybe add an antigen presenting cell using the same schema. So instead of dragging an icon in again, I'm just going to duplicate it. You can duplicate by Control-C and then Control-V to paste on your keyboard, or one way I like to do it is just hold down Alt key on your keyboard and then drag the icon. There you go. Duplicates it. And then I'm going to rotate this duplicate to face the bottom right cell and move it out a little bit like this, leaving a decent amount of space here between them to show some cell-cell interactions. And then for the top cell, I'm going to maybe make it red, like so. And at this point, you might ask, okay, so I have all these giant pieces of these background elements jutting out of the canvas. What's going to happen when I save the illustration? So that would be a great question. There is a preview button at the top right here, where if you click, you can see when you export what image you're going to get. And in fact, as you can see, the bounding area of the canvas is where it's going to cut off. So everything outside is still part of the saved illustration, but they won't be exported when you want to export an image. Speaking of save, in BioRender, because we are a kind of robust web app, we are able to save your illustrations automatically as you're building it and editing it. So you never really need to worry about losing any save progress. So for instance, watch this little note here at the top right of the canvas area. If I move this, so I made a change, saving changes, and the change is saved automatically. So essentially, you can basically just edit things and they'll be saved as you go. So at this point, one quick tip I'd like to give to everyone who attended my webinars is that when you're building a figure and you have these big background elements that kind of set the stage and you put them in and you edited them, one very helpful thing is actually to select them all. So I'm going to hold down Shift and multi-select and select them both, and then lock them here in the toolbar. And what that's going to do is, as you can see, I'm clicking and dragging and they stay there. And that way I can start working on smaller elements on top without accidentally moving them. And the lock feature is great for really anything where you don't want to move accidentally in your figure creation process. Awesome. So next, I'm going to go into the icon search bar and search for nucleus. And that looks good. So nuclear membrane bilayer. I'm going to drag that out onto canvas, move it down here, resize it a little bit. So maybe I want to show some downstream effect of this cell-cell interaction in the steps to come. And I'm going to change the icon color to fit this cell style and lock that as well. Perfect. So yeah, with that stage set, I'm actually going to skip forward a little bit. I'm going to jump out of that to sometime down the line. So as you can see here, I've also positioned the nucleus for the top cell, and on the left here, it's just a little bit of a memory cue for me to think about what to do next here. With the cell set, the next thing to think about when you want to make a graphical figure like this is to think about the major players. What are the major players involved in the story you're trying to tell in the figure? And in this case, the major players are the antigen presenting from the antigen presenting cell, potentially a MHC class 2 molecule that kind of goes to the surface of the cell, presents this red antigen to the T cell receptor, the TCR on the T cell surface. And then maybe from there, we'll have a few arrows denoting signaling cascade with certain adapter proteins, etc., to the nucleus. Okay, so that's kind of the story. And let's try to retrieve those icons. So again, going back to the icon library, I'm going to search MHC. And as you can see here, a bunch of different options. I'm going to try to find the one this one, MHC2, as you can see in the preview, TCR complex. And you'll notice it has a purple outline in the library, as well as a note that says it's a grouped icon. So what does that mean? So I'll drag it out and show you. So you notice the bounding box is now purple for this one instead of the blue of the other kind of singular icons I dragged out before. And what this means is that it is a pre-made group of individual icon pieces. And you can do two things right now. One is to customize this. One is to basically ungroup it into its subsidiary components. So if I click on group here, boom, it's going to be in its component pieces. I'm going to undo back to the group. Another, and this is a really exciting feature we added pretty recently, actually, late last year, is to enter the group and edit within the group. So as you can see in the prompt, double click to edit group. So if I double click, I'm now in the group editing mode where nothing else is editable and the group is maintained. But now let's say I want the top MHC molecule to be a bit more of a, maybe a purplish red hue like this. There we go. And then maybe for the bottom TCR, I want it to be a lighter blue like this. Great. And then if I click away and click back, it's still a group. And the great thing with the group is, you move it around, you resize it, it stays the same. Okay. So I'm going to move it over to between the two cells, rotate it to the right angle, and just quickly resize it and position it properly. Something like that. Okay. We're not going to do it perfectly this time, unfortunately. But there we go. Oh, that's pretty good. Great. So now, let's say down the line, I'm going to have a few salicylic proteins inside the T cell here at the bottom, where the signaling cascade happens and passes between them, or cascades between them. And typically, I think in immunology sciences, if something doesn't have a structure that's relevant to the story, we just denote it as a colored box with a text. So I'm going to try to create this through our dynamic shape, text shape tools. So in the toolbar, if you insert shape here, click, and then you'll be able to access a bunch of these pre-made shapes that are fully dynamic and customizable. So I'm going to go with the pill option. Click that, click on the canvas, and boom. And then with that, I'm going to, again, go with maybe adapter. I double click to enter text, and then I'm typing in adapter protein A. Like so, click away. And then I'm clicking it again. Maybe I want to format the text a bit. So in the toolbar, I'm going to bold the text. Should I italicize it? Maybe not. Here's another cool feature we added very recently. Actually, might be a week ago, where now, instead of going to here in the toolbar, fill color, changing the fill, and then border color, changing the border, like so. I'm going to undo. Now you can go to color presets here in the toolbar. And then we have a nice, I guess these are curated color presets for dynamic shapes, which applies the same border color, fill color, and text color consistently for your texting shape needs. So in this case, as the example showed, I'll do this kind of greenish grass green hue style. Click that, and there we go. I'll probably change the border width a little bit of the border, maybe to 3 points. Perfect. Put that in. Maybe I'll quickly do another one. Maybe another. Actually, you know what? Maybe I'll do a rounded rectangle protein. Call it protein kinase B. And then bolt that. And I'm going to make it a bit narrower. And you'll notice this little white dot with white circle with a dot here on the bounding box. For these particular shapes, the corners are customizable in terms of rounding. So you can go all the way to basically a pill shape with fully rounded sides, or you can do a nice kind of subtle rounding here by clicking and dragging this dot. And for this one, I'm probably going to go with maybe more of a purplish hue here. Unify the border width like so. And there we go. And lastly, I want to show here is a sneak peek, I guess, into our dynamic arrow tool. So in the toolbar, I'm going to go insert line. And I want to have a nice clear flow of the arrow from the cell-cell interaction through the cytoplasm into the nucleus. So I'm going to go with a curved arrow. Click that, click on the canvas, and there we go. And you might notice immediately that there are a bunch of controls on the arrow itself. So I'm going to make it a bit bigger like so, and then show you what they do. So I'm going to move it over. Maybe the first arrow goes from the bottom of the TCR T-cell receptor to adapter protein A. So I move it over, and I'm going to click the end of that arrow, that end node, and drag it to adapter protein A. And then, as you can see, the dark blue nodes on the dynamic arrows are basically anchor points for you to manipulate the start, the end, and the middle points of the arrow. So I'm just going to move the middle point so that it has a nice sweeping curve, like so. Move the starting point a little bit more, move the end point. And let's say I want it to be more of an S curve rather than just a simple curve. So I'm going to zoom in a bit more. The smaller, paler dots on the dynamic arrow system allow you to introduce new anchor points. So if I click this pale dot here and drag, there I've just introduced the new anchor point. So you can see there's 2 middle anchor points now. And I'm going to nudge them a little bit until they look nice. There we go, perfect. And if you want to remove an anchor point, let's say I added a third one here and it just looks ugly, simply double click and it's going to remove that. Perfect. So now I'm just going to quickly customize the arrow a little bit. Maybe I'll add a line fade here in the toolbar, and that's going to give it a bit more of a dynamic style. I can, as you can see here, you can freely customize the 2 arrowheads at the 2 ends of any line or arrow. So if it's a double arrowhead, there we go. Or maybe it's a dot to an arrow. But in this case, it's from nothing to an arrowhead. So I'm going to reapply the line fade. And another cool feature we added recently that might be helpful in certain ways as you want to maybe style, if you want to construct a figure in a certain way and then you regret the way you've made certain arrows or lines, is this line type option. So let me duplicate this. I'm going to hold down Alt and make that. And let's say initially I made a curved arrow, but later I regretted that. I'm like, oh no, all the arrows in this figure need to be straight bent arrows. What do I do? So if you go to line type here, you can actually quickly switch the mode of the arrow. So here, it's the exact same anchor points. So it's starting at the same place, ending at the same place, bending / curving at the same places. But now, as you can see, you can switch between the 3 different variants. So straight means straight lines, straight, not 90 degree, but sharp bends. Rounded lines means straight lines, but the bends are curved. And of course, lastly, curved lines are where it's a nice sweeping curve. I'm going to delete that. And yeah, so I'm just going to duplicate this by holding down Alt, maybe connecting between the adapter protein A and protein kinase B, sweeping over here like so, and then maybe removing one of the anchor points, doing that, and there we go. Does anyone have any question at this point, as I'm kind of going through some of the nitty-gritty of our in my personal opinion, very cool dynamic drawing tools. Feel free to ask in the question and answer function or in the Zoom web chat. Yeah, so I think I was going to show like placing a little bit more of the icons. So for instance, maybe in the DNA. Oh, somebody asked, how did you duplicate the object? Good question. Let me kind of rephrase it a little bit here. So let's say I have this. I want to duplicate this, or really anything on that in that matter on the canvas. One way would simply be as if you're duplicating anything else on your computer, which is if you are on a Windows system, you would do Control-C, which copies it, and then Control-V, which pastes it, as you can see here. On a Mac OS system, like an Apple computer, I'll delete that. You can do Command-C and then Command-V. So essentially the same functionality, but a quicker shortcut, and you can see the shortcuts here on the keyboard. Actually, there we go. Selection actions, duplicate, Alt plus drag. So if you just want to duplicate, you don't want to like copy and then paste later or paste multiple times. You can simply hold down Alt key on your keyboard and then dragging with your mouse. Yeah, perfect. The Control-C and Control-V, like copy paste, has its advantages. So like if I Control-C now, and I can Control-V, 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 and do a bunch of duplications at once. The Alt is when you want to hold down Alt, do a single duplication, and that's it. And then you can, I guess, of course, hold down Alt and do more duplications one by one. Okay, awesome. Yeah, so searching DNA here in the icon library, looking for one that looks good. Let's go with this one, DNA medium, dragging it into the canvas as the process goes. Relocate it into the nucleus as the end of this particular demo cycle or demo pathway, and then changing the icon color. Let's go with something a bit more subdued. Maybe this black, but then I'm like, oh, it's way too dark. What do I do? Simply go to toolbar, find opacity, click, and then adjust the opacity like so. Yeah. I'm going to do another time skip here and skip forward a little bit, as you can see here. So not much has changed, but I've fleshed out the pathway, added another dynamic arrow that is straight as one band and a short arrowhead. Again, every single component fully customizable. Added a C here in the pathway. Changed, I guess, the color of the arrows through the line color here, and duplicated a few of the MHC class 2 molecules on the antigen presenting cell here, just to kind of set the scene a little bit. So this is another quick design tip, I guess, for when you want to create a very compelling, clear but compelling graphical figure for your research. One minor thing you want to keep in mind is setting the stage, making sure the central story is clear and the focus, but then also making sure that for things like this, that for instance, the APC, the antigen presenting cell would of course have more MHC molecules, and setting them on the side there just completes the story a little bit more. So the next thing I'd like to show is, I guess, a bit of a design tip as well. So in this case, we're showing the mechanism or the signaling pathway happening on a particular scale, right? You have the two cells very big, and you can see the phospholipid membranes. A very helpful thing to show is some sort of inset to orientate the viewer or the reader. So in this case, as you can see in the queue here, I'm going to actually search for dendritic cell in the icon library. And so a dendritic cell is a type of antigen presenting cell in our immune system. So I like this one. I'm going to drag it out. And I'm also going to search for T cell. And the point of this, I'm going to shrink this a little bit, is to make an inset that helps the reader or the viewer of your figure orientate themselves and understand the context. And it doesn't have to take long. As you can see, I just drag those in a few seconds, change the icon color below to match the bigger blown up cell, change the icon color above to match the color, and then simply insert shape, and insert a rectangle, make it a bit smaller, kind of like a window, and then go to fill color and make it no fill so that it's just nice box, maybe border color black. And there we go. I'm gonna put it maybe like so, just to get the angle right. There we go. Perfect. And really what that does is I'm going to drag to select them all. It's a small inset that helps a lot in terms of, let's go to preview. In terms of orientating anyone who's looking at this figure to understand, okay, what exactly is happening. Not much work, but it really helps in conveying your science. And especially if you're presenting your research at conferences, for instance, where it's a more of a multidisciplinary conference and some people aren't really familiar with your particular subject, grounding your figures with context like this helps tremendously in attracting more audience. So just now I'm going to branch out into some of the more, I guess, power user features we have. So for instance, let's say continuing here. Actually, one thing I'm going to do is, that's okay. Continuing here. Let's say I want to use this particular inset for some other figures in the future. So one thing I could do is just, I guess, open this window, open up a new illustration in the future, and then copy it here, paste it in the other one. But instead, what you can do actually is to select it. You can choose to group it or not group it. It doesn't matter. In this case, I'll just group it just so that's easier to handle. And again, you can double click into any group to edit it and then click away to exit the group. And then if I click favorite here in the toolbar or through the right click menu, what that's going to do is it's going to save this as really essentially a personal icon of yours. And if you go to favorites tab here, you'll see that this now is a personal favorited icon. And really, this can essentially be anything. It could be this whole upper cell here with all of its components. If you want to save that as an icon, you can do that. And what that does is, let's say if I go back to gallery, I make a new illustration. And I'm like, oh, I need to add that saved visualization I did. So you go to favorites, and there it is. You can just drag it into the canvas. Voila. Going back to the gallery here. And a few other features. So another helpful thing, and we see this a lot, and when I was doing my research, I would use this too, is to be able to insert imaging, maybe micrographs, maybe some logos of our institutions for some purposes into the illustration itself as part of it. And sometimes we have multi-panel figures that involve these things where you want to piece in a chart from somewhere else and a micrograph from somewhere else and annotate them, let's say in BioRender. So what you can do is go to Uploads tab here at the left side, and you can upload images here directly from your computer. You can click here, and that's going to bring up your Windows file navigation window. But the way I prefer actually is to simply open up my folder with the images I want. Let's say there's a Western blot I made that really helps the story here. So with the image, I'll simply drag it into the canvas. And it says upload an icon, give your icon name. Western blot. Actually, I think that one was not a Western blot. I think it was a Ponceau red stain. And then upload. And what that's going to do is it's going to automatically save that as a image-based icon in the Uploads tab, as well as place it into the canvas. And last but not least for today, in the canvas view at least, we also have a protein data bank integration. It's one of our first, I guess, integration plugins in BioRender. We're planning for many, many more. Some ideas include PubChem, potentially a graphing tool like GraphPad. We're not sure yet, but for now, this is, I guess, what we started with. So if you click PDB, it's going to bring up a PDB viewer. And here you can, if you work with 3D protein structures or like research that involves structural biology, this is the place for you to basically integrate that into your figures if you need. So you enter the PDB ID that's relevant for you, and then simply adjust the structure and color to suit your needs. And then you can convert to BioRender icon. We are actively iterating on features like this. Here we go. Just drag it in, and it's placed. So one exciting thing I'd like to, I guess, secretly announce here in this webinar is that we're really excited to release in the very near future a new version of this PDB module that might be interesting to some of you. With much richer functionality and much better looking proteins. So I'll delete that. So I think at this point, I'd like to just kind of go through some of the more miscellaneous things, and maybe some considerations as well. So one thing we're really proud of as a company at BioRender is that we have a dedicated in-house medical and science illustrator team who are working every day to expand our icon library as well as our template library. And the main way we actually drive this content growth is through user requests. So if you see here at the bottom left, there is a request a custom icon button. If you click that, for any user who is on an appropriate subscription plan, one of the huge benefits is that you can actually request a custom icon. And how it is that you can fill the information here, as you can see. And one of our professionally trained medical illustrators would actually fulfill the icon request and create it and let you know, and then you can use it in your figures. And additionally, a really cool thing is that you're also helping out with the scientific community at large, because we would also include that into our icon database, or icon library rather. And other scientists in your similar field who might be using BioRender or may come to use BioRender in the future would simply be able to access that icon and use it. So I think I mentioned early in the introduction, but so at BioRender, I'm working mostly as a user experience researcher. So what I do is I try to understand and talk to different scientists and see how BioRender can be improved to fit their needs. And one of the things that I notice actually is that with lines and arrows, users and even scientists who are really experienced with BioRender tend to try to use arrows or lines that are exactly what they think they need, but without realizing that you can actually fully customize at any point to whatever you need. So for instance, let's say for a particular figure, I want to have a two-ended arrow that's curved. So looking here, the only two-ended arrow is this bent double arrow. So if I add that to the canvas, I might go, oh, okay, I have the two arrowheads, but this is a straight bent arrow. I can't use it. I need it to be curved. So there's a few different solutions. One is to go to line type, change it to a curved line, and voila, you can do that. Another option might be to add simply a curved line and then add arrowheads, as you can see in here, to both ends, and now you have a curved line with double arrowheads. Another cool feature we added that might be helpful for some of you who might be doing some cyclical diagrams, such as something to do with plasmid arrows or plasmid vectors, or some workflow that is cyclical, or some pathway that has cyclical elements, is these arc arrows. So there are specialized arrows that don't really behave the way that most of our dynamic arrows do. So I'll move over to the side a little bit here and show it here. So if I insert an arc arrow, looks pretty similar. I'm going to make it bigger. But what's different is that when you drag the ends of the arrow here, doesn't matter where my mouse moves, it's actually going to go along a rail, like so. And let's say, what if I have a few breaks here where I need to have text or icons in between to show the steps? Well, in that case, if you hover over the midpoint of any of this arrow segment, you see a scissor icon, and you can just click to break it. And now, if you click any of these now broken arrow segments, they're still on the same rail, so you can simply adjust them as you need. Make the space maybe for your steps, and then maybe here I need 2 steps, I'm going to click that. Maybe here I need to have a break here instead of in the middle, so I'm going to click and hold the scissor and drag, and there we go. And if I click away, you might notice already the first click I did to break this arc arrow dimmed everything else, and that's because it automatically converts it into a grouped icon. So if I click away, click back, as you can see with the purple outline, it is a grouped icon. So you can double click to continue editing this. Or you can click away, select the whole thing, and maybe apply line fade to everything, you know, the usuals. Change the stroke width, maybe make them more of a broad arrowhead, maybe make the broad arrowheads a bit smaller. There we go. Change the color. Yeah, so I think that'll wrap it up for today, for our webinar today, in terms of running through some of the functionalities that I think will be helpful for you to make your first figure and hopefully subsequent figures great. I definitely missed some things, but of course, you know, please do sign up and jump into the app and yeah, explore on your own time. Some of the features might be really relevant to you particularly. And I guess, yeah, lastly, I'm going to go back to the gallery page, go to BioRender templates. You can see this on your own time, of course, when you sign up. You can sign up for free without entering anything like credit card information, anything like that. So it's completely no obligation. And you can browse all the icon library in the canvas view or the templates here outside in the gallery view, check things out. And when you click use template here, in the template page, it simply creates a new illustration with this as a starting point. And you can, again, fully customize to your needs. Going back to gallery. Okay, so I'm going to hop back very briefly just to wrap up our presentation today. So obligatory talking about free accounts as well as some subscription options if you're interested. So if you do enjoy the free forever version of BioRender when you sign up, and you would like to, let's say, publish a figure that you've made in BioRender, or make a lot more figures, or if you'd like to make expedited icon request or anything like that, we do have a variety of different subscription plans for you to choose from. You can, of course, find more detail at the URL spelled out below, biorender.com/pricing. And also just a quick mention, if you are a graduate student, that is master's up to postdoc, or if you're working with someone who is a graduate student, we do have an exclusive student plan at a discounted rate of just $15 US per month. And feel free to contact us in the support channel, I guess, if you're interested. Yeah, so that wraps up our webinar for today. Thank you everyone for joining. A few little housekeeping things. If you want to review this webinar or share the webinar recording with a peer of yours, we will be providing a recording in a YouTube playlist within the next day or so in a follow-up email from this webinar. So look out for that. And of course, if you have any other questions, please feel free to reach out to our amazing support team at support@biorender.com, or you can reach me personally at jerry@biorender.com. Thanks, everyone. I'm going to leave the webinar session up for a couple more minutes. If anyone has any last questions before you move on to the rest of your day, I'll be happy to answer them in the question and answer or in the webinar chat. Thank you. Have a great day and great rest of the week. Bye-bye. Oh, Samen, you brought up a great question, actually. How to export figures created in BioRender. I should have touched on that. I'm going to quickly jump back into here. So what you can do is you can export either through the export button at the top right here in your canvas view of any illustration you've opened, as you can see, to access the export window, or you can go to File and Export Illustration. And then here you can specify the file type. Maybe for PNG, you want a transparent background. For PowerPoint, and then the resolution. And then when you export, it's just going to generate an image, as you can see here, that's automatically downloaded into your system. And then you can use that for whatever you need. Thanks for asking that.